Everybody remembers our plan for lab today. If you were here last week, we're going out first on the soil pit description field trip. We're going to meet in the lab at 1:30, load up into the van, depart to the steam plant parking lot, where we will arrive probably around 1:45, 1:50. If you're joining from a class late, you can either meet us in the ag sci parking lot or in the steam plant parking lot to hop in the van. We'll be planning to be back to campus around 3 or 3:15. At which point you guys will be good to go. Everybody who missed last Thursday's lab activity, you will have finished the lab activity in lab, and then we will load up and go out and do the soil pit description field trip second. You'll be back to campus around 5:15, 5:20. Sound good with everybody? I have some exciting plans to share. I'm gonna sit down for this.
Okay, it's serious.
Okay, I'm really excited because I have a fun opportunity. I think, for context though, I'm in my 4th year of doing my PhD. I've been paid on like a half teaching, half research contract this whole time. So teaching is like technically part of my job, but it doesn't really count towards like the research progress that I'm making. So I have to write like a research paper and chapters in the dissertation that teaching doesn't really contribute towards. But that's not really ideal for me because this is what I want to do. Like I want to pursue a teaching career in academia. I want my dissertation to be like uniquely tailored or like help set me up for that kind of career that I want in the future. So I've come up with a way to hopefully integrate this teaching experience with my dissertation and my research project by doing a research project, which I describe as follows. I have to read the script here. Okay. So study title: Effect of Answer Key Feedback on Student Confidence in Soil Description. The researchers are myself, University of Idaho SOIL Research PhD student, Michael Decker, a employee of the U of I College of Graduate Studies that works over there, and Dr. Zachary Taylor, my advisor, associate professor in the Soil and Water Systems Department. Premise: as students in Soil 4540 biology class, you will gain experience completing technical descriptions of soil profiles. Students in past semesters have commonly asked me during these field trips, are my descriptions correct? However, soil descriptions involve subjectivity sometimes, and there is not always a clear right answer. Some descriptions can be definitely wrong, some may be more commonly accepted than others, but even expert soil scientists disagree with each other all the time during soil descriptions. This ambiguity of correctness can be frustrating to students. That is why traditionally, I have not made an answer key description for class soil pits, but instead have a big picture discussion at each soil pit of the overarching takeaways and notable features. Answer key description itself would just be my opinion about some of the soil properties, and it's not reasonable to expect anyone at all to perfectly match my descriptions. That said, the positive feedback when your answers as students agree with mine could be beneficial for your learning. I want to investigate this. What this means for you is when we go out on field trips, I'm going to split the class into 2 groups that will remain constant for the entire semester. One group will complete the soil descriptions and have a big picture discussion with me afterwards, as is the traditional method for this class. The other group will complete the same soil description, but review afterwards by having an answer key description read aloud by either me or Logan, our lab TA. I will still be available to the answer key group to have the big picture conversation after. Everyone will be doing the same work for lab and be graded by participation, which is exactly the same in the groups and as all past offerings of this course. The only difference is that the answer key group will receive more granular feedback. What you will do to collect data for this project, I will use surveys published through Qualtrics. If you choose to participate, you'll be asked to rate your confidence in your soil description skills, both as a whole and for each portion of the description sheet. There will be some additional questions about your perceptions of soil science and your learning, as well as open-ended items if you wish to elaborate. Completing the survey will take up to 5 to 10 minutes. These surveys will be sent for you to take at 3 separate times over the semester. Is this mandatory? No, you are not required to participate. You can skip any question or stop at any time. Your choice to participate or not will not affect your grade or standing in the course at any time. Are there risks or benefits? I anticipate no more than minimal risks to you. While no discomfort, stress, anxiety, or other emotional reactions are expected from the surveys, keep in mind that you may stop at any time. There is also no direct benefit to you for participating in this survey, though your honest participation may improve the course experience for future students. Are answered confidential? Yes. Surveys do not ask for your name, student ID, or any other direct identifiers. To link your surveys over time, you'll create a private code from simple prompts. Only the research team will see de-identified data. Results will be reported in aggregate and no information will be traced back to you. If you have questions, you can ask me anything at any time. You can also contact my collaborator, Michael Decker, at mdecker@uidaho.edu if you prefer. For questions about your rights as a participant in this research, you can also contact the University of Idaho Institutional Review Board, IRB. I don't know if this is going to work. I'm excited to try it though. It's very real that you guys, you want to know, am I doing this right? Are my answers correct? But whether or not you say something slightly sticky, and I said it's moderately sticky, like you don't deserve to have like 0 out of 5. Oh, I got all the stickiness wrong. You know, like I think that would be probably harmful in you guys' ears, but if you get some of the stickiness right, then maybe that's competency that this correctly. So there's no right answers. So surveys are completely anonymous. It will not affect your grade at all. Everyone's graded the same way. My plan is to send out the first round of surveys today after lecture that hopefully before going out on this field trip, so we have like a baseline, your confidence going into SOIL pit descriptions. We'll do it again partway through the semester, and then one more time, identical survey after the last SOIL pit field trip. So we're a pretty small class. I don't expect to have like any groundbreaking findings or publish this in a paper or anything, but the main idea would be either to repeat this next year and then go somewhere with it, or maybe write this up as a bigger proposal, maybe get like 10 university pedology classes involved. 5 of them do answer key descriptions, 5 of them not. Everybody takes the same surveys and go from there. But this is like the pilot data. You guys are what I can face more of this research. So thank you for your participation. Happy to take any questions you have about it now. Otherwise, keep an eye on your emails shortly after lecture or a survey link from Paul Tietz. It'll be email from me with a link to the Paul Tietz survey. And hopefully it won't take long. You can skip any items you want or the open-ended ones, but your honest feedback is greatly appreciated for me and my learning and my research on how to best be a social science instructor. So thank you for your consideration, your interest, your participation. So that. Do you guys want to know the groups? These are randomly assigned. Starting off with the answer key group will be Gage, Gabby, Isaac Lenz, Tegan, Daniel, Drake, Amelia, Lucas, and Sophia. Everybody else, which is Isaac B, Catherine, Harrison, Brian, Verity, Josie, Ryan, Ethan, Sloan, and Gabe will be the no answer key group, the traditional experience. I don't expect either one to be better than the other. I consider them equal treatments. I'm just curious to know the effect it'll have, if any, on you guys as you're learning SOIL description. With that, we can continue our lectures on the soil forming factors with hopefully 2 today. We'll have time to get through 2 new ones. To review once again, we're talking about CLORP, the soil forming factors. CLORP, our acronym stands for Climate, Organisms, Relief, Parent Material, and Time. Organisms and relief are the 2 we are setting out to cover today. Once again, these soil forming factors explain why the soil looks the way it does, whereas the soil processes we learned about before are explaining how. I, in general, prefer talking about and thinking about the soil forming factors because because of these effects that they have on soil, you can see them, like they explain all the differences. They explain the differences in the soil classifications that we have and all the soil properties that we see in all these different landscapes. You can travel around when you go to different areas, you see the effects of these different factors. Meanwhile, those four processes are happening everywhere, and they explain how these soil horizons and these soils develop, but the soil forming factors tell the story, tell the why of why any soil looks the way that it does. We reviewed climate last time. Now for organisms, let's get rid of this. Our organisms include plant community, the plants growing on the surface and interacting with soil, macro and mesofauna, so insects, invertebrates, animals, and all the way down to microbial scale. Really many different scales of organisms interact with any given soil. This figure here is a representation of like a humid, temperate zone soil. So not necessarily everywhere, but the relative abundance of these soil organisms per square meter of surface area in this humid, temperate zone, from relatively large, large animals, macrofauna, scaling down to protozoa, microflora, bacteria, fungi, algae, single-celled life forms that become more and more abundant as their size decreases. This scale here is a nematode and a collembola or a springtail, as if they were perched on the head of a pin for scale reference. And some say that if you took 1 cubic meter of soil, these springtails or collembola specifically, you have 100,000 or more in just 1 cubic meter. Some people say there's more organisms in a gram or two of soil than there are humans on Earth. So what is that to say? Soil's full of life, teeming with life. We like judging the soil and describing the profile, and even the soil is judging us back. It's alive. Who's to say? And it varies across environments, of course, but there's an abundance of life in soil that's doing things. What effects do organisms have on soil material itself? They're responsible for a lot of the production and the decomposition of organic material. This production decomposition especially ties into nutrient cycling, so between carbon, between nitrogen, maybe phosphorus even, transitioning from like plant available forms to be incorporated into biomass and then decomposing and being mineralized back into plant available forms. Maybe it's respired into the atmosphere and then brought back somehow. Maybe it's eroded into the ocean and then, in case of phosphorus, brought back really slowly over a long time. But organisms, when we're thinking about carbon and nitrogen especially, have a really important role in the nutrient exchange and the economy of nutrients in a soil profile. Organisms also affect weathering in soil, so maybe creating more soil in the first place from like repeated action over a long time. So the physical interaction of roots with like bedrock, for example, over a long time, that physical interaction of those roots can break down rocks, but also more specifically like the root exudates. So the organic acids maybe that come out of roots and the CO2 respired by roots helps break down either whole entire rocks like our horizons or the specific minerals that make up a horizon, and increasing the weathering effects, creating more of these secondary soil minerals that we are going to talk about in a couple weeks coming up, mineralogy for pedology. And then organisms also create a microclimate of like a shading effect. We talk about solar radiation effect on soil or temperature. If you think of a forest or maybe sometimes single a single tree as possible, creating a different microclimate environment affecting how the soil behaves, functions, or maybe even looks. So we talked about or let's see, how about interaction, or like an example case study of effects of organisms on soil profiles. Focused on, let's see, we're going back to the 1930s here. Near the city of Los Angeles in San Dimas, the city of Los Angeles was once upon a time looking for ways to get more water in their drinking water municipal reservoirs. Even back then, California was concerned about droughts and water shortage and water availability. And looking around at all these reservoirs in like forested watershed areas, some of these city planners in Los Angeles were wondering what it would take to convince the forests that they don't need as much water as they're taking up. What if we could get more water in the reservoirs by, I don't know, maybe changing the tree's water demand, or getting more water out of these forested hill slopes? And there's really no way I can think of doing that except for changing the trees themselves. So that's what they did. A large section of the forest was cut, and these huge lysimeters essentially were built, like a big plot of soil hooked up with a lot of plumbing, so like drainage ditches around it to measure the water that's running over the surface and also infiltrating through the soil profile. Measuring rainfall runoff, infiltration, percolation for 32 separate plots. Essentially, big trenches 11 feet wide, 21 feet long, and 6 feet deep were cut between these to measure the infiltration of these different treatments that they were going to set up. Thankfully, this could have gone a lot worse. At least they used native vegetation, native pines and tree species if they were bringing stuff in from all over the world. Thankfully, they weren't going too crazy, but California scrub oak, coulter pine is what they used. Unfortunately, fortunately for the experimenters here, the soil was just a little too disturbed after all this construction that it didn't really represent natural soil drainage conditions. There were shrink swell, created like macropore channels that water would just run right through that didn't really represent the natural soil condition. Also, they lost all the soil structure too. So think about granular versus subangular blocking of natural soil profiles, how that's gonna affect water infiltration rates. All the structure was lost here. So their experiments didn't really work necessarily. However, these plots were essentially a blank canvas of soil exposed to the same vegetation, the same relief or slope conditions, the same parent material, the same age of formation, but the only difference between these plots were the tree species they planted on them. So this ends up being a really good example of a biosequence here. So forest cleared, replanted, and then their study was compromised by all this heavy soil disturbance. Although they failed to discover a way to improve the water use efficiency of a whole entire forest watershed, they did set up a really good biosequence or comparison of effects of organisms on soil over time. And in these pine plots, this is 80, 90 years later after it had been set up, some soil scientists followed up with this experiment here and dug some small soil pits in these constructed plots. And where they had pine growing, observed shallow A horizons, really barely anything up there at the top. Not much in the way of an A horizon. No real soil structure still after 80 or 90 years. And also no earthworm activity. Whereas these oak plots had deeper A horizons, these golf tees you can see here, soil scientists might use those to delineate where they put horizon boundaries. So our first A horizon, maybe a little slightly darker color here, is at about 10 centimeters. Maybe slight changes in the soil structure down here or something about it for these second and third horizon layers. We had deeper A horizons, structured development observed, maybe BWs, and also many earthworms. So why this difference? I think the acidity of the pine needles and the litter of these colder pine plots kind of prevented the same biological activity that was able to recover in these scrub oak plots, especially with those earthworms that cycle nutrients, break down carbon, help restore that granular structure maybe at the top, or at least some subangular blocky first to improve infiltration. And with the earthworm burrowing and casts, improves the nutrient availability well, just building up soil horizons in the first place. So I find that as an interesting consolation, a silver lining to big investments of infrastructure back in the day in the 30s when water was pretty valuable in the Dust Bowl era, but I guess people needed jobs and they got some trenches dug in, and soil scientists made something come out of it, at least. Consolation. A more familiar or closer to home, maybe thinking about just grassland organisms, the effect of deep rooting prairie grass on soil. Through these deep roots, a lot of carbon storage and carbon storage created by this below ground biomass. I defer to the range majors of the room. 80% of total biomass of grass species that sound reasonable would be below ground compared to what's grown above ground. These dryland grasses, Palouse adjacent at least, maybe North Dakota or similar, above ground carbon per square meter around 3 to 4 times lower than the below ground carbon storage, thanks to all this root activity. Numbers, not so important, you can't even see them on the screen in here, but just that ratio, I think, is really notable. And it's the same in other environments too, even the alpine, high elevation Swiss Alps, similar kind of ratio, 300 grams of carbon per square meter above ground, and almost 4 times that below ground because of these productive roots and their ability to store carbon in the soil. Deeper A horizons, right? Like we talked about, increasing the vegetation productivity through increased precipitation or moisture availability, increases the depth of the A horizon, dark color of the soil profile, good A horizon development because of this root activity. Probably not too surprising, pretty intuitive about the intricacies of deep rooting grass species. On the Palouse, that's been known here for well over 100 years. The study of local vegetation from 1917, really kind of setting up the high below ground productivity, a lot of root interacting with the soil. But the roots themselves have the important interaction, like physically with the soil, in addition to just being a source of carbon, but creating the granular structure, exuding chemical compounds, and this zone of interaction here that we call the rhizosphere, maybe a 2 millimeter zone just around diameter around roots in the soil. And there are whole entire soil scientists that make their careers in this rhizosphere here. They're studying the interactions of plants and the soil, these feedbacks, how the nutrients flow, how water flows, how plants are affecting their soil in this rhizosphere. Zooming in on some of these roots, we notice that maybe it's not only the roots themselves, but also some symbiotic relationships going on. Mycorrhiza specifically are fungi that are symbiotic with plant roots. The idea behind this, plants are providing sugars from photosynthesis, right? They have the above ground, the leaves, and they receive the sunlight, photosynthesize sugars and nutrients that they provide to the fungus. In return, the mycorrhiza provide water and nutrients that they are better at extracting from the soil than maybe the root or the roots themselves are physically able to. So the fungi specifically, sometimes will penetrate into the root cells. I remember from my botany class in undergrad seeing like the thin section slides of the mycorrhizal fungi, arbuscular mycorrhizal fungi, as it's known in the business, like colonizing into the plant root cells, provide the water and soil nutrients while receiving the sugars. But they don't have to incorporate with the root cells themselves. There's also ectomycorrhiza that just remains outside, covers the surface of the plant roots. Still has this productive exchange. We've also got plants with root nodules, the legumes or leguminous crops. In these root nodules, there are bacteria in addition to the fungi. The mycorrhiza, these symbiotic bacteria are able to split gaseous nitrogen, N2, which is a really stable inert form, like 80% of our atmosphere. But using sugars provided by plants, these rhizobia in these root nodules split that N2 unavailable to plants and then produce ammonia, leading to amino acids, nucleotides, other mineral and plant available forms of nitrogen. So really important biological function. Plants provide the sugars, bacteria nodules fix that nitrogen, biological and fixation. And this is really useful for rotation agriculture. So leguminous crops dating back to ancient Egypt had been used in rotations with other crops to maybe restore some nitrogen into the soil. A lot of some of my coworkers in my research lab are doing this kind of research, but on intercropping systems. So what if you're growing like barley or peas in the same field at the same time as wheat or some other crops? Is this nitrogen fixation interaction beneficial to all the plants around it, or is the competition too much, or is it just not practical? Is really what I can't get around is the manual harvesting of all these plots, because it's a single row of barley and a single row of peas, and there's just I'm not totally sold on intercropping agriculture, but that's what they're doing their research on to figure out a lot of this stuff and the value, the use of it, I think. But still important benefits and uses of the ability of plants and organisms within those plants, within the soil, to keep these nutrients moving. Soybean roots are a great example of that, those nodules. Okay, now kind of getting on a smaller scale or more broad geographic scale, we've got lichen with driving some soil processes here. Lichen is a hybrid between algae and like fungal cells, or fungal DNA, I guess, that really enables lichen to grow nearly everywhere. So on in the tundra, hot and dry deserts, rocky coasts, toxic slag heaps, even inside some rocks and between mineral grains, you can have lichen starting to grow. Lichen are not really parasites on plants, even though it kind of looks like that sometimes, maybe if they're growing on a tree. They just use anything they can grab onto as like a growing substrate. What lichen are really good at is accelerating the weathering. So they're exuding these organic acids, and they're adding organic material to these maybe blank slates of ecosystems, like a primary succession kind of deal. They're the first biological colonizers. And yeah, they start the ecological colonization. If your goal is to prevent weathering of something, let's say a big granite sculpture, or Mount Rushmore specifically, the national park at Mount Rushmore invests in power washing all the lichen off of George Washington and all those other presidents because lichen accelerate weathering. That's what they do. And if you don't want any noses breaking off, you gotta. Step one is keeping the lichen away. Lichen are also interesting indicators of air quality. They have like those diatoms maybe in the past we talked about with the Alpha horizon. Some lichen have a very narrow range of acceptable environmental conditions, and if air quality is decreasing or there's some pollution or contamination, lichen may be among the first indicators of environmental quality. So that's a lot of general effects of like the whole grassland ecosystem or a forest ecosystem, maybe a field of soybeans and lichen across a landscape, but can there be a single plant effects on soil development? And yes, for this example, we traveled to New Zealand. If only Gabby was here, our New Zealander in class. The classic example of a single plant effect on the soil is this kauri tree native to New Zealand. They're super tall, they're super wide, super slow growing, really old, kind of rivaling the giant sequoias and the redwoods of California, but southern hemisphere version. These kauri trees have really acidic litter that facilitates leaching in the soil and also kind of outcompetes a lot of other plants from growing around like the base of these trees because of how acidic litter it makes, which really leaches out nutrients from the soil. And we are familiar with this nutrient leaching from any given soil horizon, where we observe these E horizons forming directly under these trees. And then a BHS, like the development of a spodosol on these really local scales. They call these like baskets or egg cup podzols, this podzolization process happening here. Around the world, though we call them spodosols, other countries know them as podzols, kind of created on a really single area local scale. So Paul McDaniel, the former pedologist here, has spent a lot of time in New Zealand. I think got a sabbatical or two there, and these are his examples. I would love to check out this kind of custom-made E-horizon at some point in my career, should I be so fortunate. But I'm also excited to share today that Paul McDaniel himself will be coming in to give a guest lecture or two while I am traveling to the Soil Science Society of America conference. This will be in November, but you will get to hear from Dr. Paul McDaniel himself on a couple of lecture topics. Really excited to bring him in again. So questions about organisms, effects of just biology on soil development. No questions. Water relief. Our next soil forming factor, we got plenty of time probably. The consideration for relief in the way that climate can be like the dominant soil forming factor. So orders I think of like aridosols formed in a really dry climate. Organisms can be the dominant soil forming factor on like histosols, maybe are all organic material. Mollisols too have show a lot of effective organic activity, and that is like the delineator of that horizon, the central characteristic of it. We don't really have that same thing for relief. You don't have a relief dominated soil profile, but relief is really important for explaining the differences in soil profiles that are maybe relatively close together. Comparison, so we had a climosequence and a biosequence for climate and organism factors. Comparing soil across different landscape positions or with different relief inputs would be known as a topo sequence or catena. A lot of soil scientists will talk about a catena of soil pits maybe along top, middle, bottom of hill slope as an interesting comparison. Relief is still a very important for us to describe. It's kind of hard to tell just by observing a soil profile what annual precipitation is, what the annual temperature is, right? There's challenges to measuring soil temperature in the first place. But relief is a little more tangible. You can, and on our field sheets, we will be describing the percent slope, the aspect, hill slope position, and also slope shape. So a two-dimensional and a three-dimensional representation of, or characterization of the relief we're dealing with here. Starting with slope. Slope is really just a steepness measured in percent. This is pretty straightforward. Think of it as a rise over run. So let's say over a horizontal length of 100 feet, the change in elevation is about 7 feet. That would be 7% slope. 7 feet of vertical gain over a 100-foot linear distance. We measure this by our clinometer. So shooting from 2 points of equal height above the ground, we're measuring on the percent side. Clinometers measure in degrees and percents. Make sure we're using the percent side. To really just measure the steepness. Second is the aspect, so the compass direction that a slope is facing. Aspect is really valuable for determining solar radiation that affects the soil. Solar radiation is like the more for temperature, right? And varies across the landscape depending on your angle to the sun. So in this figure, we have equal cross-sectional areas of sunlight. So starting out with the same, I guess, square units of sunlight, depending on the angle of the ground surface, depends on or determines how much that same solar radiating energy is distributed. So think of it like your shadow at noon versus your shadow at sunset, which is going to be a lot longer. When sunlight is distributed over a large area, like you're sloping away from the sun, you have less energy per area of ground surface. Over level surface, this is kind of the intermediate angle here, or distribution of the radiation, versus if you're at like a right angle to the incoming radiation, you get the maximum efficiency heating. So in the northern hemisphere, at least where we are, the south aspect will always receive the most sun. Sun's going to be in the south. This would be a south-facing slope receiving the most radiation. The north-facing slope away from the sun, be cooler, probably wetter. We'll see some examples of this. If we travel to New Zealand to describe some of these kauri tree spotozols, we would have to keep the opposite in mind, that it's the south face is the less sunlight, north face is the warmer one. You can see an example of this really anywhere you drive. It's a bit hard to miss when you start looking for these, but any kind of hill slope, or this is the canyon behind the Lewiston Grade, for example, Hatway Canyon. The south-facing slope, so this is the north side of this little canyon, but it's the direction that faces south, receives more sunlight or more solar radiation over a smaller area, relatively warmer, relatively drier, it dries out sooner in the coming out of the spring maybe. And it's also, we see prairie vegetation. We got grass growing over there compared to the north side, less sunlight, it's cooler temperatures. That helps moisture retain in the soil and enough for trees to grow and get forest vegetation examples from side to side. I have only ever driven by this canyon, but if we could find a place to park and maybe zip line across it or something and have a soil pit comparison, a little topo sequence here, Katina across the canyon. This is what we might see. The south-facing aspect on the left has south aspect on the left. We got this deeper A horizon calcification as a process going on because it's drier. Maybe the same amount of total rain is falling on both sides of the canyon, but because of that solar energy, it's drying out sooner. Calcification as a process, It's happening everywhere. It's just the annual depth of wetting is going to be shallower for the south aspect. It's not going to get as deep. The carbonates are going to be able to precipitate out into this effervescence BK horizon. We don't see that on the north side. So calcification as a process is happening in this one, along with that deeper A horizon from the difference in vegetation. Whereas the north aspect is going to retain more of its moisture, maybe have an increase in the lessivage or that clay alluviation process happening. You get a deeper BT1, BT2 horizon, maybe deeper soil, as well as a result of that, more water, more weathering, not quite as shallow to this basalt bedrock is what we got. What we have here, capital R, present on that. Okay, last two things for relief are the hill slope position, described as summit at the top, shoulder, you start to curve down, back slope at this part, foot slope you're evening out, and toe slope is kind of as you're moving away from that hill slope, it's really easy to, or starting with a summit at the top and working our way down. Summits are generally stable, generally a good place for soil development to happen. Some exceptions might be loose hills where summits themselves have been taken down a couple notches from soil erosion. But in general, and most other places where you have maybe soil that's inherently more resistant to erosion, the summit is a good place for soil development. Shoulders are going to be a little more shallow as we're starting to have these erosive processes going on in general. Backslope positions dominated by transportation maybe over the surface or through flow, water flowing rather than down, like straight down from the soil profile. They're going to be flowing down due to gravity, which is kind of maybe parallel to the ground surface a little bit more. Through flow going on with the back slopes. And then foot slopes dominated by accumulation now. Could be any combination of water from flowing downhill. It's going to start collecting at the foot slope. Any solutes that it brings with it, anything, nutrients or anything that's dissolved in the soil solution, going to start accumulating at the foot slope, as well as mineral soil itself, if it's been eroded from the summit or the shoulder, carried down over the back slope, going to perhaps begin accumulating at this foot slope position. Also, none is an option. If you don't have this idealized standing on a slope and you know exactly where you are on this simple slope here, none is also a valid hill slope position when you're just not associated with a hill, if it's a really flat area, or if you're in a different position like a floodplain on the landscape. A lot of times a floodplain won't necessarily be a foot slope or a shoulder or anything. Floodplain would just get none for a hill slope position usually. This is great, and this is really easy to think about when we have a simple slope like this, but the natural world is not as straightforward all the time, as you can imagine. There might be a general scale summit and shoulder and back slope and foot slope, but within that, you can have a little more fine interactions here where you're pretty close to the summit of the hill, but it's a relatively steep grade in between two kind of flatter portions. It's like a local back slope on a general shoulder, and the one you pick to describe it with, sometimes the professionals will go both. They'll kind of hedge and say it's a local foot slope on a regional back slope or something. As long as you do something that makes sense and is also consistent, and also that you can evaluate consistently and consider reasonably, I think, for determining what is actually affecting the soil at this location. So the intro soils labs, soil 206, field trip destination of the old Arboretum. You got two soil pits pretty close to each other. One we say is like a local back slope because it's a 30% or more steep slope. The other one is more of a foot slope, even though that's also kind of steep at 10 or 12% slope maybe. They're both very high on the hill. If the definition of your hill is from the golf course to Paradise Creek, that would be the local shoulder, but or the general scale shoulder, and the detailed scale or the local back slope and foot slope. That's what we decide to describe it with, because that effect of runoff from the surface on the back slope and starting to accumulate on this relatively flatter area affects or creates the differences that we see in those two close by slope pits. Okay, and then slope shape is our last feature. This is a little more complicated, where we had a 2D, uphill to downhill drawn before. This is now a 3-dimensional consideration, where this at the top of the figure here, imagine that being an uphill, like a flat upland region, and like a valley or a lower elevation here at the very bottom. And this kind of notch in the middle here is like the valley between them. So high elevation here, and you're going down a pretty steep slope. You can be at this head slope position, maybe at the head of the valley, where water is going to flow inwards. It's a little more concave of a shape, where at the start of this draw, essentially, and then at the nose slope, at these two positions out further, the slope is kind of curving outward. It's more like a convex shape. So these concave versus convex versus linear is how we describe slope shape for any soil description place. We evaluate linear versus convex versus concave in 2 directions. So starting on the left side here, you will notice that each one of these representations is flat going from side to side. So it's linear flow in the horizontal direction. And then this middle one is the convex example, where it's kind of bending outwards versus concave sloping inwards. And then it can be the same in different directions too. So we're linear going across, or linear going straight down again, but convex going across. That would be a linear convex slope, where it changes the convex flow path of the water. Really, the best example of this is where you have a convex. It's convex both going top to bottom and from side to side. Water is going to be flowing very divergently away. This would be the nose slope example, where water spreads, versus concave in both direction. Convergent flow is going to be the important scenario, where water is collecting maybe at the foot slope position on the very bottom. That does it for relief. We are good to go. I will see you guys in the lab or in the steam plant parking lots.